Researching protein-protein interaction is indispensable for understanding physiologic processes in cell biology field. In this video, we introduce on-membrane digestion technique, which allows the researchers to perform a comprehensive analysis of binding proteins by allomeres in a convenient manner.
The main advantage of this technique is that we can improve the throughput of protein mix analysis for crude immunoprecipitants, because separation of proteins by polyacrylamide gel for electrophoresis is not necessary.
Begin conjugating the antibody to the magnetic beads by mixing the anti-GFP antibody with the beads in 500 microliters of citrate phosphate buffer. Rotate the mixture at 50 RPM for 1 hour at room temperature. And then wash the conjugated beads 3 times with citrate phosphate buffer containing 0.1% polysorbate 20. Lyse transfected cells according to manuscript directions. Scrape the dish and transfer the lysate into the 1.5 milliliter test tube. Then clear the lysate by centrifuging at 15,000 times G for 5 minutes and collect the supernatant. Prepare unlabeled magnetic beads by washing them 3 times with 500 microliters of citrate phosphate buffer. After the final wash, remove the buffer and add cell lysate to the beads. Rotate the mixture at 50 RPM for 30 minutes at room temperature. Perform magnetic separation on the magnetic stand and collect the cell lysate to bind the target proteins to the